Bismillah ar-Rahman ar-Rahim. I will continue about cellular reaction to an injury. Lecture 3 includes apoptosis and intracellular accumulations. What is apoptosis? It is a falling off. It removes cells no more needed by the body. It is a suicidal program, affects single cells. Mainly it is physiological process. It could be and rarely it is a pathological process, and it is not associated with inflammation as in necrosis because the plasma membrane in apoptosis remains intact and the dead cells are rapidly cleared before the leakage of cellular contents to elect an inflammation, and it is an ATP dependent process. So it affects single cell, while necrosis it affects groups of cells. It is mainly physiological, while necrosis it is a pathological process. It is not associated with inflammation, while necrosis it is associated with inflammation. We have a physiological apoptosis, or as we said, mainly apoptosis it is physiological. Apoptosis occurs in many normal situations to eliminate harmful cells and cells that have outlived their usefulness. Examples: the programmed destruction of cells during embryogenesis, involution of hormone-dependent tissues upon hormone deprivations, such as endometrial cell breakdown during menstrual cycle, cell loss in proliferating cell populations, such as in intestinal crypt epithelia in order to maintain constant number. Also, eliminating cells that have served their useful purpose, such as neutrophils in acute inflammatory response and lymphocytes at the end of immune response. We have a pathological apoptosis. Rarely we have a pathological apoptosis. Apoptosis eliminates, or pathological apoptosis remains cells that are genetically altered or injured beyond repair, and does so without eliciting a severe host reaction, thereby keeping the extent of tissue damage to a minimum. Examples of pathological apoptosis: DNA damage following radiation, cytotoxic anti-cancer drugs, hypoxia, and hypoxia can damage DNA if repair mechanism cannot cope with the injury. The cell triggers intrinsic mechanisms that induce apoptosis. Accumulation of misfolded protein because of mutations in the gene or because of intrinsic factors. Excessive accumulation of these protein in the endoplasmic reticulum will stimulate apoptosis. Certain infections, particularly viral infections, in which the loss of infected cell is due to apoptotic death, that may be induced by virus, adenovirus, and human immune deficiency virus, or by the host immune response such as in viral hepatitis. Atrophy in parenchymal organs following obstruction of their ducts, parotid, and pancreas. So, lastly, atrophy in parenchymal organs following obstruction of their ducts, such as in the parotid gland and pancreas. This is liver, and this is the apoptotic cells or apoptotic bodies. This is the example of a pathological apoptosis. This is a viral hepatitis showing the apoptotic cells or apoptotic bodies. Now it comes into the mechanism of apoptosis or pathogenesis of apoptosis, and we will speak about the role of mitochondria in apoptosis and in general in cell injury اللي هو necrosis and apoptosis. As we said, in case of necrosis, there will be mitochondrial damage with decrease in the ATP, with increase of reactive oxygen species, and this will lead into multiple cellular abnormalities with necrosis. While in apoptosis, there will be a decrease in the survival signals, DNA, protein damage, and this will increase the pro-apoptotic proteins with decrease in anti-apoptotic proteins. This will lead into a leakage of mitochondrial protein with later on apoptosis. The plasma membrane in case of apoptosis remains intact. That is why there is no inflammation associated with apoptosis. This is the morphology of necrosis and apoptosis. As we said, this is the normal cells. This is the cell which undergo reversible cell injury, which shows myelin figure, swelling of endoplasmic reticulum, and mitochondria with membrane blebs. Whether there is a progressive injury, there will be a cell death and necrosis. While in case of apoptosis, this is the normal cell. There will be a condensation of a chromatin with membrane blebs, cellular fragmentation, and later on it will be phagocytosis of apoptotic cells and fragment. And this is the phagocytosis of the apoptotic cells by histiocytes. Now it comes into intracellular accumulations. Under some circumstances, cells may accumulate abnormal amounts of various substances, which may be harmless or associated with varying degree of injury. Intracellular accumulations could be in the form of lipid, and we have two types, either as a neutral fat, we call it a fatty change or fatty degeneration, or in the form of cholesterol, we call it atherosclerosis, deposition in the wall of blood vessels, we call it atherosclerosis, or in the form of a protein. Visible protein accumulations are much less common than lipid accumulations. Example, nephrotic syndrome in the kidney, in which there is heavy leakage of protein across the glomeruli, giving the appearance of pink, hyaline, cytoplasmic droplets. Another example of intracellular accumulation, glycogen. It is excessive intracellular deposition of glycogen are associated with abnormalities in the metabolism of either the glucose or glycogen. Example, in poorly controlled diabetes, glycogen accumulates in the renal tubules, cardiac myocytes, beta cells of the islets of Langerhans. Other type, pigments of intracellular accumulation. Pigments could be exogenous or endogenous. We will speak about it. And calcium in the form of calcium deposition. So intracellular accumulations could be in the form of lipid, protein, glycogen, pigments, or calcium. We will speak about the intracellular accumulations of lipid. There is a lipid law. All lipids are yellow grossly and washed out clear microscopically. The fatty change refers to an abnormal accumulation of triglycerides within parenchymal cells. Most often is seen in the liver because, as we know, the liver deals with the metabolism of fat. It may also occur in the heart, skeletal muscles, kidney, and other organs. What are the causes of fatty change? Could be due to toxins, protein malnutrition, diabetes mellitus, obesity, and alcohol abuse, which is the most common cause of a fatty change. Grossly, or gross features of a fatty liver: the liver enlarged, yellow, soft, and waxy. The liver enlarged becomes soft and waxy. Histologically, this is the site of fatty degeneration in the liver. It appears as intracytoplasmic fat vacuole, and it is most prominent around the central vein. This is the site of a fatty vacuoles. Mostly it is around the central vein. This is the site of the fatty degeneration. Now, the appearance of cholesterol crystals, or it appears as a clefts-like or slit-like in the tissues. This is the appearance of cholesterol, slit-like or a clefts-like in the tissue. Deposition of cholesterol occurs in blood vessels, causing narrowing of blood vessels, causing ischemia, and the disease we call it atherosclerosis. Intracellular accumulation of calcium. We will speak about pathological calcification. We have normal calcification. Normal calcification are found in bones or teeth. But we will speak about pathological calcification. We have two types. The dystrophic calcification, when there is a deposition of calcium in necrotic or dead tissue, abnormal tissue, with a normal serum level of calcium. This is, we call it dystrophic calcifications. The example of dystrophic calcification: deposition of calcium in advanced atherosclerosis, in damaged heart valve, and in the tumors. This is a damaged heart valve in case of a chronic rheumatic endocarditis with calcification of the valve. We have another type of calcification, pathological calcification. We call it metastatic calcification, in which there is a deposition of calcium salts in normal tissue with hypercalcemia. There is an increased level of calcium in the serum. This is a metastatic calcification in the lung. Most common sites of a metastatic calcification here in lung and the kidney. What are the causes of hypercalcemia? Increased secretion of parathyroid hormone due to a primary parathyroid tumor, destruction of bone, example, immobilization, or tumors such as multiple myeloma or leukemia associated with increased bone catabolism. Hypercalcemia may follow vitamin D intoxications, or lastly, renal failure, in which phosphate retention leads to secondary hyperparathyroidism and so into hypercalcemia. Common sites are the kidneys and lungs. Pathological features. Gross features. The calcium appears as white, fine granules, chalky-like, or it forms a clumps which may be converted later on into a stone-like. Or could be a the histological appearance of pathological calcification, cellular and or extracellular basophilic deposits. So, the histological feature of pathological calcifications or calcium deposition in the tissues appears as cellular and or extracellular basophilic deposits. This is the appearance of calcium histologically. This is dystrophic calcification in the wall of the stomach. Now it comes into other intracellular accumulations اللي هي pigments. What do we mean by pigments? They are a colored substances. We call it pigments. We have an exogenous pigments coming from outside the body, and we have an endogenous pigments synthesized within the body itself. First, we will speak about the exogenous pigment. The most common exogenous pigment is carbon, and the best example is coal dust, the air pollutant of urban life. When inhaled, it is phagocytosed by alveolar macrophages and transported by lymphatic channels to the regional tracheobronchial lymph nodes. Aggregates of pigments blacken the draining lymph nodes and pulmonary parenchyma, giving the disease we call it anthracosis. So this is the anthracosis. This is anthracotic pigment in macrophages in the lymph node. These are the pigment. Tattooing, another example of exogenous pigment in which there is an intradermal inoculation of a color substances. Intradermal inoculation of a color substances, we call it a tattoo. Now we comes into endogenous pigments, and first melanin is brown black pigment produced in the melanocytes located in the epidermis. It acts as a screen against ultraviolet light. Excess melanin deposition may occur after exposure to sunlight, Addison disease, adrenal gland failure, or in pregnancy. We call it cloasma. And this is the appearance of melanin. This is the appearance of melanin. It is a black-brown pigment. Second endogenous pigment, we call it lipofuscin, or pigment of aging. It is insoluble, brownish, yellow granular, intracellular, and it accumulates in the heart, liver, and brain. When lipofuscin pigment presents in large amount with atrophy of the tissue, we call it a brown atrophy. So, lipofuscin pigment, when present in large amounts with atrophy of the tissue, we call it a brown atrophy. Lipofuscin, it is a pigment of aging. And this is the appearance of a lipofuscin. This is pigment in the liver. This is the appearance of a lipofuscin pigment in the liver. Third example of endogenous pigment, we call it a hemosiderin. It is a hemoglobin-derived granular pigment. It is a fine golden yellow to brown pigment. It accumulates in tissues when there is local or systemic excess of iron. Example هو البروزي اللي هي الكدمة. This is the hematoxylin and iosine stain in the liver, and this is the appearance of hemosiderin. And this is a special stain. This is the hemosiderin for iron. We call it a Prussian blue. Lastly, other example of endogenous pigment, it is the bilirubin. It is the end product of heme degradation, and elevated serum bilirubin causes yellow discoloration of skin and sclera in cases of jaundice secondary to liver diseases such as hepatitis. This is the appearance of bilirubin. Thank you.